When I was some time ago in Senegal, I came across someone who told me that.
I just been in the pharmacy and they told me I have hypertension. I don't know what to do.
Huh. So, is it that bad?
Medicines are expensive. And they told me treatment will be for the rest of my life.
Oh, that's annoying. So what will you do?
I really don't know.
Perhaps I have a solution. Let me explain. You know, hypertension has become a problem that's very common not only in wealthy areas, but also in tropical countries, low and middle income countries, as I will show you on a map. You see, now it's also very common in sub-Saharan Africa. And the problem is how to provide a treatment that needs to be taken for years, for the whole life, very often, and could be accessible. So we had to devise a way of finding that sort of treatment. How could we do? When we arrive in a village like this one and try to find a solution for a very common disease, we start with a survey. And when we did this survey, we found that for this particular disease, there were many, many different treatments used. Because they don't only use the standard treatment, they also use anything they've been, perhaps they heard about something from their neighbor, their grandparents, the traditional healer, and they try it. But then the question they asked is one of these many treatments better than the others? Is there something we should all use or we should recommend? So how would you do that? We devised a method to try to find among hundreds of different treatments which one is the best one. I'll show you how we did that. The usual discovery process is to start with a lab, as you have here on this side, and then if you find something that seems interesting, you will go on with animal studies, and then clinical studies with humans, and at the end, you will start to distribute, to spread this to the population. We thought that in this case of trying to find one effective, safe treatment in a population that use many different things, we couldn't go that way because it would take years to study all these treatments. So we did the other way around. That's why we called it reverse pharmacology. We started with a population survey, asking the people about what they do and which outcome they've got. In the case of blood pressure, what did they use? What did they take as a product or any other treatment? And then when they would measure their blood pressure again, was there any difference? Was it improved? And then we get a big data. It's actually a sort of big data the local people have made through the years and the generation. And with statistical analysis, we can see if there is some particular treatment associated with a better outcome. So that's what we did. And in Senegal, we found that two common plants were associated with some good outcomes. So we would first look at what are these plants. And in this case, they are both food products, means they are very safe. The risk of having undesired effects, secondary effect, is pretty low. So we could go on with the next step. So the next step would be to use these plants exactly as they are in the local recipe. So both actually are drinks which are often taken for breakfast. Hibiscus is this red drink which we also have here and is used in many places in the world. Bissap, or it's called karkade, in North Africa, in Middle East as well. So the next step was to do this randomized control trial, which means we take a group of people who have the same disease. They all accept to be part of the study. They are divided by random in 2 groups. So these 2 groups will be very similar in terms of all known and unknown factors of blood pressure, characteristics, and so on. And then each of them will receive a different treatment, and we compare the outcomes. In this case, it was a little bit more complicated, but with the same principle, is that we started with 5 different groups. You see here, the line on the top is the upper figure you have in blood pressure. You have always 2 figures, systolic, diastolic blood pressure. And we had, for each plant, 2 types of preparation. That is the local recipe, a tea, and then we also tried a powder of the plant prepared in the form of tablets or capsules for the 2 plants. And at the right, this square was the control group. They had the standard treatment, in this case called captopril in Senegal. And we followed the patients for 6 months, and at the end of the 6 months, we could see what was the progress in terms of blood pressure. And you see that all the groups had a very similar progress, which means that they were equivalent. After that, the next step was to go to the lab. And there, we found, in the case of hibiscus, I show you here, 3 different mechanisms of action and active substances that were responsible for these mechanisms, which were actually the mechanisms that are already known for standard anti-hypertension drugs. One is the captopril, which is the standard in Senegal. The other one was the anticalcic, another group of antihypertensive drugs. And the third one was diuretic. That means it helps urinating, so lose the excess, eliminate the excess water. With that, I should also present you the sort of group we have for that sort of study, because you need a very multidisciplinary group. In this case, you had the professor here in the middle, with whom we devised and developed this method. And on this side, you have his doctoral students in medicine, pharmacy, phytochemistry, and then the traditional healer who would provide the recipe. And I must add that in terms of intellectual property, the traditional healer decided that he would give his recipe to humankind. So we were in a copyleft system, open source situation. And on the other side, you have the lab technicians, and the very last one is the foreigner who helps coordinate the whole thing and have also the products going to all the labs in the world for the study of the mechanism of action. As a result, we end up with a plant that has been studied in terms of safety, efficacy, in the same way as a standard drug. So all this to explain you why I could tell to this person at the end, well, I think there is a plant you could try. It's cheap and available, so perhaps it will help you.
Thank you so much, doctor. I go to the market and look for it. And now another example of decentralizing science. Once I was in Mali and I see this.
Oh, I cut my leg. It's a bad cut. It's a dirty wound, and I don't know what to do because I went to this hospital, but they have no disinfectant. They have nothing, and then it might become worse, and I am afraid maybe they will need to cut my leg at the end. What should I do?
I know this is a frequent situation. In many countries, health centers have no access of disinfectant and neither drinkable water. Let me explain you. I have a solution. Oops. You take kitchen salt, water, you mix them together. Then, the salty solution, you put inside this nanowater device, which you connect to a power source that could be solar. And then, through an electrolysis process, the salt and water molecule will be broken, and then it will form the chlorine solution, a really potent disinfectant that you can use it. And you know what? The same disinfectant can be used for making drinkable water. One volume like this is enough to make 320 liters of drinkable water. But since the process lasts only 23 minutes, you can do several times per day, and then get drinkable water for 1,000 people per day. One device, 1,000 people. You know that according to the World Health Organization, about 2 billion people drink contaminated water, and about 500,000 die every year because of it, because of diseases as diarrhea, cholera, and many others. Is it acceptable that still today 500,000 people die because of it, because having no drinkable water? Is that because we don't have a solution? We have a solution. Is that because it's expensive? No, it's low cost. Is that because it's complicated? No, it's just salt, water, and then that's all. How many decades should we still wait to make drinkable water as a priority?
So you have seen 2 examples of decentralizing science, that is participative science.
Science that responds to the needs of people.
To the essential needs.
The essential needs of everyone.
Even the poorest.
Even the most deprived, the most underprivileged.
The idea with decentralizing science is first ask the people about what they need.
What they need and what they do.
What they do and what they want.
And
then, if you discuss with the people the question that could be answered with science,
then you find solutions that are accessible, affordable, sustainable. Solutions that comes from the field and goes back to the field.
So once we have got a solutions, but it's not yet widely known, the next challenge is disseminate.
Thank you.